by definition, we're going to call an antigen. By definition, an antigen is a substance that triggers an immune response. But now we know really what's happening at the level of cells is it's triggering a response that starts with the T cells. Either you get a T helper, T cytotoxic T cell all jazzed up, and it kills the cell, or what triggered the killing of the cell was the antigen. The antigen molecule triggered this. And in the case of the helper T cells, this, if it functions as an antigen, is going to get the T helper cell all jazzed up as well. Okay? All right, let's go back to our system here. We know that the T cells, specifically the T helper cells, can pick up these antigens from other cell types, right? They could also pick them up from other places as well, but one of their favorite places to go are these phagocytic cells because they've got all kinds of interesting peptides to look at. So here is this cell right here. It's some kind of a phagocytic cell. It's a macrophage. The macrophage brought in the bacterium, chopped it up, and these little pieces were then presented on the class II molecules. This little piece was then checked by the T cell receptor of a helper T cell, and it got interested in it enough that it takes it. So now you got a T helper cell wandering around with that little antigen stuck in its T cell receptor. Okay? Now, in MCB, I talk about T cell receptors and B cell receptors. I like the analogy, forgive me if you've heard this before, but basically the T helper cell and the B cell can interact with each other, and they interact through their receptors. The T helper cell picked up this antigen from one of these phagocytic cells, and now what it wants to do is go find a B cell to show it to. My analogy in MCB is to describe the helper T cell as like a 6-year-old boy and the B cell as an adult. And basically what happens is the T cell takes that antigen that it has bound to its receptor and starts showing it to B cells. Now, one of the places you can do this a lot is here, because this is where the B cells hang out. So the T helper cells are showing their antigen to as many different B cells as they can find. The B cells all have different shaped B cell receptors. Right? That's one of the things we do is make a whole army of B cells that have different shaped receptors. So one of the things that T cells do is try to find a B cell with a receptor that fits that piece. Now, if it picked up a peptide from a phagocytic cell, chances are pretty good that it's foreign protein, right? That's the only thing that it should be able to pick up anyway, but it picks up this foreign protein and it presents it to a B cell with a certain shaped receptor. And that B cell, maybe its receptor doesn't fit the antigen, in which case the T cell wanders off to find somebody else, some other B cell that will recognize that peptide. For me, it's like taking a 6-year-old boy to the park. They pick up a stick, they bring it to you, and they say, wow, look at this stick. Is this the coolest stick you've ever seen? And you're looking at the stick and you're going, dude, it's just a stick. That 6-year-old boy, we haven't finished the process yet. Until somebody shows some interest in that stick, it isn't over yet. So he shows it to you, you're like, dude, it's just a stick. He wanders over to somebody else, shows it to him, it's just a stick. He finds somebody else until somebody shows some kind of interest in that stick. At which point, Timmy drops the stick and he's done with it, and he goes off and finds a rock and does the same process all over again. Little Timmy, the helper cell, wanders around trying to find a B cell that will get excited about this antigen. If the first few he tries don't get excited about it, meaning they don't bind it, he'll just keep trying. There are lots of B cells in a lymph node. Keep trying until you find one. Eventually he finds a B cell that gets all jazzed up about this interaction here because its B cell receptor fits that. It fits it. And once that happens, we get this jazzed up B cell. Okay? Little Timmy found the B cell that says, wow, that's the coolest stick ever. And once he's found that cell, this process results in mitosis of the B cell. One of these cells stays as a B cell, and one of these cells becomes a plasma cell. And then these cells proliferate to produce more B cells and more plasma cells. Plasma cells from plasma cells, B cells from B cells. Now, the difference between a B cell and a plasma cell is simply this. It's the location of this receptor. B cells keep the receptor locked in their membrane as a B cell receptor, whereas the plasma cells secrete the receptor. And the secreted receptor goes by another name called an antibody, and it goes by another name called an immunoglobulin. Anytime you see glob, think protein. So this is an immune system protein, and antibody meaning it's anti-something else. So the plasma cell secretes antibodies and immunoglobulins. Okay, so far so good? All right, so the macrophage picked up something, maybe from that little twig that came into the skin. The macrophage picked one of these up, chopped it up, and presented a piece of it on that MH2 class receptor. It was picked up by a helper T cell that eventually found a B cell that recognized that peptide. That B cell then went through a mitotic event to produce a B cell here and a plasma cell, and then this B cell will go through cell division to produce more B cells. This one goes through cell division to produce more plasma cells. Okay? These B cells that result from this process are often called memory cells. And I'll come back to this because this is an important part of acquired immunity, is whenever we mount a response against a particular shape, we have cells that remember that shape into the future, as well as a group of cells that actively produce the antibodies. Okay? All right. Now, this is all happening at the lymph node, right? We have this macrophage charging up a T helper cell, which then interacts with a B cell to produce memory cells as well as plasma cells. And then these secreted B cell receptors, called antibodies, these receptors then end up exiting the lymph node, collected in the lymph, right, and then entering into the circulatory system. And before long, these antibodies that recognize that shape are going to end up all over the place. They're going to end up in the circulatory system, get squeezed out of more capillaries, and end up in more interstitial fluid, and then back. So they begin to circulate throughout the body. The idea is you should be able to recognize particular shaped antibodies by taking blood from somebody. You would pick up these antibodies that were produced in response to a particular shaped peptide that triggered this process. So just as an aside here, there's lots of talk about antibody testing for the COVID issue to try to figure out how many people have actually been exposed to the virus versus how many people are showing a response to the virus in terms of fever, shortness of breath, or any other symptoms that we associate with COVID. If somebody has been exposed to the virus, at some point one of those viruses got hit with a macrophage or a mast cell or somebody else that chewed it up, presented it, got the T helper cell excited, presented it to a B cell, and finally found a B cell that recognizes that shape on the surface of COVID, and now you've got memory cells plus these circulating antibodies. The presence of these antibodies in somebody's blood indicates that at some point in the past, a macrophage picked it up, and the only way it could pick it up was if the virus were in the body to begin with. Okay? So antibody testing is all about trying to figure out who's been exposed to what. Okay? All right. Okay, now, when you're exposed to an antigen, think you're exposed to a particular pathogen. The pathogen exposure is what triggers this whole response, but because the pathogen had to be chopped up in little pieces first, and then the little pieces are what trigger the immune response, we talk about exposure to an antigen, but what we really mean is exposure to a pathogen. It's just the antigen is what molecularly triggers the process. Okay, so here, relative amount of specific antibody in the blood. Here's weeks after first exposure to antigen A. So it took a few days for us to mount an immune response. All right, looks like my battery is low, so I'm going to charge it back up, and then I'll see you on the other